Hello everyone, today I will introduce to you my second topic, which is cell cycle genes regulation dynamics revealed by RNA velocity and deep learning. Everyone here knows that our bodies built by primary structure named cells, and during our life, a cell divides into many new cells through a fundamental process called by cell cycle. Despite the fact that cell cycle is an important process of life, the genes regulations dynamics still unclear, and thanks to the development of single-cell RNA sequencing and methods to analyze and splice RNA space of cell cycle-related genes, the order, including the first order is Henry Riba, and the corresponding order is Nacho Molina. They developed a deep learning tool named Deep Cycle to describe a high-resolution map of cell cycle transcriptome. And in this study, the authors applied Deep Cycle to identify major ways of transcription in stages of cell cycle in different cell types, including embryonic and somatic cells. First, I will introduce to you the cell cycle genes regulations. The primary of the study based on cell cycle genes regulation. The cell cycle has 4 periods. The first is G1 phase. Cell will spend most time in this phase to do living activity, in which a lot of genes are transcribed into RNAs, then the RNA is expressing their proteins, and in this period the cells prepare things that needed for the division of cells, so that the volume of protein and organelles increase so much. Next, the DNA will be duplicated in S phase here, and after that, in the G2, the second G phase, this phase involves further cell growth and organizations of cellular contents. And finally, for normal cells, for proliferation cells, cells pass to the mitotic entry commitment point, pass the checkpoint here, and then they will attain into M phase, in which from old cells will divide into 2 new cells. And deep into the molecular pathway, there are a lot of genes related to cell cycle were found in many various studies, but the quantitative picture of tight dynamic regulation of gene expression is incomplete. So the author did this study to comprehend the cell cycle genes regulation using single-cell RNA sequencing data and RNA velocity. And all you here know about single-cell RNA sequencing, but what is RNA velocity? This is a method to generate an accurate capture cell cycle dynamic based on genetic data. It means that this method will capture all the image of movements of some targeted genes in cells during a period that we call cell cycle. And so, how does RNA velocity do? The RNA velocity indicates the transcriptional state of individual genes based on their percentage of nascent and mature RNA. We know that eukaryotic genes have intron and exon, and only exon encode the protein sequence. So after the transcription, the nascent mRNAs, in other words, the unspliced mRNA are needed to alternative splicing to make the major mRNA, or we call bispliced RNA. So the RNA velocity based on the relative abundance of unspliced and spliced RNA to calculate the distribution of unspliced uh, and spliced risk across uh, all the cells, and this distribution called by unspliced spliced RNA space, and from this it is possible to estimate if a specific gene is being transcribed or inactivated in any single cells, and the dynamic of this gene can be simulated like that. And there is a typical workflow that usually used starting with a set of risks is converted to the matrix here with cells and genes derived from spliced and unspliced mRNA molecule measurements. The matrix are processed to generate phase plot describing a dynamical transcription process, and finally the transcriptional dynamic effect extrapolated and displayed in a low-dimensional embedding. In this study, the output of RNA velocity used as the input for deep learning methods called deep cycle. And thanks to the big data created from RNA velocity, a deep learning approach could be a good strategy to have a deep insight into genes, regulation of cells, and to analyze the fully circular RNA velocity pattern of different cell types with high performance in different cell phases. Deep cycle was developed based on neural network, a neural network assigning an angle. In other words, it is transcriptional phase. I not here, and the result of this cycle is the circular structure. It describes a transcriptional stage of a gene of its cells, and the deep cycle was trained by a dataset including over 530 samples of expression of unspliced and spliced RNAs of genes in a dataset here, and the deep cycle was compared with other current use methods such as cyclone Rebelio or cyclone, and so its advantage in performance and flexibility in applications for different cell types. In this study, deep cycle was applications in two arms. The first one is the recapture specific cell cycle dynamic in different cell types, and the second is to characterize the branching point where the human fibroblast exits from the cell cycle. So let's jump into this research. The other one to study the dynamic of cell cycle. So the research focus on proliferating cells. The measures of public sequencing RNA sequence datasets have been generated to study the overall population of cells in a given condition, and they contain heterogeneous cell types. So the author compared three different populations of proliferating cells, including mouse embryonic stem cells, ductal cell progenitor, and human fibroblasts. they culture the other culture cells and generated a single cell RNA sequencing library. And then they found that the human fibroblast divided into two subpopulations, and only one subpopulation expressed cell cycle genes. So the other first focus on this subpopulation of human fibroblasts, and the single cell RNA sequencing data show the RNA velocity pattern with total spliced and unspliced reach of ductal cells are only 8 million, while the data of fibroblasts are 210 millions, and the data for mouse embryonic stem cells are 20 millions. We can look at the figure E. These three populations of cells have the similar fractions of unspliced reach, and on the dataset contains genes with a circular pattern in the spliced and unspliced reach space in accordance. And the cycling genes, the researchers expect that the cycling genes will be characterized by fully circular pattern as they complete both the activation and deactivation phase. And to overall, these datasets constitute a unique opportunity to study genes regulation throughout the cell cycle in different mouse and human cell types. And next. And to comprehend the activities of regulation genes during S and G second GM phase in cell cycle, some marker genes are check the expression in single cells, and because the transcriptional phase does not contain information about cell cycle phase transitions, phase markers are needed to identify the different transitions through the cell cycle. For control of deep cycle, the author did an analysis with the detection of cell cycle phase by Z-score. They counted the number of RNA per cells, then they calculated the Z-score expression, and based on the average Z-score per cell, the author defined the transition, and the conditions are used to differ cell cycle phases. I show here, in which a cell in M jump into the first G phase, since the first pin across the transcriptional phase when RNA count per cell drops. A cell from G1 phase change to S phase when it is saw the first pin across the transcriptional phase after the maximum cyclin E score, and finally a cells from S phase change to second G phase when the theta where the G2M score becomes greater than the S score. And using the deep cycle to analyze RNA velocity of those marker genes, the deep cycle indicates the transcriptional phase, which are normalized between 0 to 1, and then the transcriptional phase are aligned such that mitosis occur at angle equal 1. We can see here the path of the cells around the cell cycle can easily be identified in the two-dimensional projections, And the results show that the mouse embryonic stem cells have the lowest number uh, of cells in G1, while the ductal cells and human fibroblasts have much more extended G1 phase. We can see that only 19% of mouse embryonic stem cells in G1 phase, but in contrast, there are up to 79% and 59% cells in G1s for ductal and human fibroblasts, respectively. After that, they will compare results from deep cycle with detection of cell cycle phase by Z-score, and it is found that deep cycle have the same result of control. The author concludes that deep cycle allow a genome-wide investigation of gene expression dynamic across the cell cycle. And to predict cell cycle core transcription factor, deep cycle used with a linear model to infer transcription factors activities from the single cell RNA datasets, and I will call the transcription factors as TF for short. The motif analysis predicts that most TF activity takes place in G1 phase when the cells need to decide whether to go into another route of replication or to address the cycle in order to accomplish a new function. We can see the result here. There are a lot of TF placed at the marker for each cell cycle phase, and among the most significant activities, some TF factor were identified. The first is YY1. The YY1 targets are upregulated in the G1 phase in all the datasets. It suggests a general role during the cell cycle. And I have a question here. Why they suggest a general role during the cell cycle in all the datasets? And the answer is its function. The YY1 is known to induce proliferations and maintains pluripotencies of mouse embryonic stem cells, and is bind to chromosomes during mitosis, and its transcriptions start already in the second GM phase, suggesting pioneering activity at the beginning of the new cycle. For both mouse embryonic stem cells and ductal cells, the E2F2 families play as important family protein, and this family are known to act at the beginning of the cell cycle, specifically for the G1 first GS transition. And to E2F related motif activities peak in between the J1 and the S phase to activate the genes necessary for the transition. And other factors seem to act similarly between mouse embryonic stem cell and ductal cells regarding to the ductal cells. The other found a very high correlation between the YBX1 mRNA level and the activity of its motif, where both are constantly increased from G1 to M phase. And The YBX1 is known to positively regulate the G1 and G2 M phase of the cell cycle. And about the factor appearing in the human fibroblast, similar to YBX1 in ductal cells, the mRNA level of FOXM1 or FOXM1 rose constantly from G1 to M as expected by its function during mitosis. And in the figure C, we can see that the mouse embryonic stem cell maintains the pluripotent state, and it is really crucial necessary. Among the genes related to this process, the strongest activation happens for the targets of STAT, TCF3 and POU5F1, which are enriched in the second G M phase. To overall, the undifferentiated cells show a strong and unique wave of activation of transcription factors in G1 phase, and in contrast, the more differentiated cell type, the activity activities of transcription factors across the G1 phase cluster into two groups. The first, we can see here, the first group display an early activation directly after mitosis, while the second group here is display a late G1 activation. The other believes that ways of transcription factor link with the cell phase decisions, and they will investigate this opinion in the next experiment. And using the deep cycle method, the research characterized a subpopulation of human fibroblasts with low cycling activity, initially excluded from analysis. The full fibroblast population was found to comprise with over 70% in GO1 and 15% in S phase and only 9% in second G M phase. These were validated by flow cytometry analysis, and some G1 cells were observed to be as big as S, and second G M cells suggest that an increase in size for cells in GO and G1, they are a little bit unclear the re-entry into the cell cycle. The fibroblast population were divided. We can see that were divided into two subpopulations around mid G1 with markers suggesting the non-proliferative cluster might include cell transitioning into the GO phase. The study implemented a method to infer paths connecting cell states in two-dimensional space, revealing the trajectories with the highest cell densities. The GeoMarker genes were checked and results support to the hypothesis of non-proliferative cells being at an early stage of quiescence. And the expression analysis indicate that non-proliferative fibroblasts might represent a differentiated state with the uh, FOSM1 down-regulated. It is known as a marker for quiescence, and the CDK2 activity control the proliferation quiescence decision consistent with CDK2 mRNA expressed in fibroblasts. The genes regulating cell proliferation were down-regulated or quiet in non-proliferative fibroblasts. We can see here a lot of genes plays regulations role in cell proliferation. And to overall, the deep cycle enable to characterize the geotransition of cells in wild-type fibroblasts without the perturbing the cells and revealing candidate genes and transcription factor regulating quiescence. In summary, the author developed deep learning methods called deep cycle, use the results from RNA velocity to describe the transcriptional stage of a gene in each cell via the transcriptional phase, an angle called the transcriptional phase. And the deep cycle proved effective in different cellular models across organisms and developmental stages, as well as identify specific transcriptional factors in each cell populations. And it's also found observed single cells and their characteristic of mRNA level existing the cell cycle in single cell RNA sequencing sample. In future plan, the author want to develop deep learning to provide more comprehensive insights into transcriptional phase distributions and unspliced RNAs. And the deep cycle also can apply to review dysregulated cell cycle mechanism and potential rock development targets in further study. Thank you for your attention.